Let's continue with part 4 of the fungi lecture. Fungi play very critical roles in the carbon and nutrient cycles of all ecosystems globally. They're critical to the functioning of ecosystems in two different ways, associated with the carbon, nitrogen, and phosphorus cycles. First, fungi are the primary organism breaking down dead animal and plant materials. In fact, they are the only organism capable of breaking down the lignin within trees. Second, fungal hyphae interact with plants in a symbiotic way to extend the ammonium and phosphate nutrient uptake zones for roots within the soil, basically creating larger root networks. So fungi are a key component of the carbon cycle. That is, if photosynthesis is the dominant process in plants, whereby CO2 is reduced to form organic matter, producing biomass, when that biomass dies, it is the fungi which are able to decompose the cellulose and lignin into smaller components to be taken up by bacteria and other organisms residing in the soil. And at the same time, fungi form a symbiotic relationship with plants. We call those fungi mycorrhizal fungi, and they're associated with nutrient uptake in a symbiotic fashion. In one part of the partnership, the plant provides carbohydrates or food to the fungus for growth. In exchange, the fungal hyphae extend out and take up both nutrients and water for plants. We call those fungi mycorrhizal fungi, and they play a very critical role in nutrient uptake. They are of two types, the ectomycorrhizal fungi, shown on the left side, and the arbuscular mycorrhizal fungi, shown on the right side. They both serve the same ecological and adaptation function of taking up nutrients, primarily nitrogen and phosphorus and water, for the plant at distances far from the root. They differ in their morphological interactions. On the left, you can see ectomycorrhizal fungi, which form a sheath surrounding the root tip, penetrating inside the root, in between the cell walls of the plant root. From that point, the hyphae extended outward into the soil, and they form a very dense, continuous sheath layer around the root. In contrast, arbuscular mycorrhizal fungi penetrate in and connect to the plasma surface, the plasma membrane of each of the different cells. So they've gone beyond the cell wall onto the plasma surface membrane, but they do not penetrate into the plant cell. And again, the hyphae extend outward into the soil for the purpose of taking up nutrients. Two different approaches, the same symbiotic interaction. The advantage of a mycorrhizal interaction has to do with the fact that most nutrients are far from the root. In the process of taking up nutrients near the root, and those nutrients are primarily things such as sulfate and phosphate and ammonium, a nutrient depletion zone is created. Basically, it's a zone in which there are no more nutrients. So how can a plant get nutrients that are far from where that root nutrient depletion zone is? The answer is by using mycorrhizal hyphae, which are long filamentous structures, as you've now learned, which extend out into the soil and allow the hyphae to pick up nitrogen and phosphorus and transport it back to the root. This happens in a process which should be very familiar to you now. In order to function, the hyphae need a carbohydrate source. The plant provides that carbohydrate. In return, a different form of growth occurs. That is not growth towards the food source, but away from the food source into the open soil. Here, enzymes are once again excreted by the hyphae, and those break down organic matter which is in the soil, allowing the hyphae to take up ammonium, peptides, amino acids, and phosphate, all of which are then used for plant growth. This is a symbiotic relationship, a mutually beneficial relationship between the fungus and its tree or plant. Mycorrhizal associations allow a plant to grow faster and larger, providing essential nutrients at a faster rate, simply because the nutrient zones surrounding a root are very quickly depleted. In classic studies, we see that plants grown with mycorrhizal fungi versus without mycorrhizal fungi are quite different in size. In fact, some plants will not even grow without a mycorrhizal association. One of the consequences of sterilizing soils are that the fungal spores in the soil are killed. So plant growth on sterilized soils is often very limited until the soil is inoculated by adding fungal spores. Fungi also form another kind of symbiotic relationship. This is a symbiotic relationship between a fungus and a cyanobacteria, or a fungus and an algae. That combination comes together to produce a lichen. A lichen is a symbiotic combination of a fungus and a cyanobacteria, or a fungus and an algae. The nutritional growth, or the carbon source for the fungus, comes from photosynthesis by either a green algae or a cyanobacteria. They have very different forms and shapes, as shown here on these images. Lichens are extremely slow growing and may take decades before they're actually visible to the naked eye. You can get an idea of just how slow lichens grow by looking at tombstones in cemeteries. The older the tombstone, the larger are the lichens that are growing on the tombstone. Now let's stop and test your understanding of what you just learned with these 3 true-false questions. After you have written down your answers, restart the video to see if you have the answers correct. Here are the answers to those questions. Each and every question was true. That completes this portion of the lecture.